Hello everyone. Hope you are all doing well. Welcome to our PBL project. Let's begin by going over what the PBL project is. The PBL project, or the Project-Based Learning Project, is a student-centered, real-world project that involves students working together to solve a problem or answer a question. PBL projects are made to develop critical thinking, problem-solving, and decision-making skills. So, what was our project about? Our project was based on photosynthesis. This right here is what we call the poster board. This single slide contains all the information regarding our experiment. To get a better idea of our project, let's take a look at the abstract. Photosynthesis is the primary biological process by which green plants, algae, and certain bacteria convert light energy into chemical energy which is stored in glucose. This process, essential for life on Earth, occurs in the chloroplasts of plant cells, where chlorophyll absorbs sunlight and drives the conversion of carbon dioxide and water into glucose and oxygen. Photosynthesis can be broken down into two main stages: the light-dependent reactions and the Calvin cycle. In the light-dependent reactions, light energy is absorbed by chlorophyll, which then uses this energy to produce ADP and NADPH. These molecules fuel the second stage, the Calvin cycle, where carbon dioxide is fixed into glucose. Oxygen is released as a byproduct during the light-dependent reactions, making photosynthesis a critical process for maintaining atmospheric oxygen levels. In addition to its importance in energy production for plants, photosynthesis is foundational to ecosystems, supporting food chains by providing energy to herbivores and higher trophic levels. It also plays a vital role in the global carbon cycle, helping regulate atmospheric carbon dioxide levels and influencing climate patterns. Ongoing research is focused on improving the efficiency of photosynthesis, particularly in crops, to address challenges related to food security, climate change, and environmental sustainability. Now, let's move on to the purpose of our experiment. The primary objective of this experiment is to observe the production of oxygen as an indicator of photosynthetic activity in plant cells. This experiment uses spinach leaf discs submerged in a solution containing bicarbonate, providing carbon dioxide, which is necessary for the light-independent reactions of photosynthesis. When exposed to light, the spinach leaf discs undergo photosynthesis, producing oxygen as a byproduct. The oxygen causes the discs to become buoyant and float to the surface of the solution. This provides a direct and visible means of measuring the rate of photosynthesis by monitoring how quickly the discs float in response to light. Another purpose of this experiment was to identify the different pigments present in spinach leaves, which play crucial roles in the absorption of light for photosynthesis. Plants rely on various pigments, such as chlorophyll A, chlorophyll B, carotenoids, and xanthophylls, to capture light energy from the sun. Each pigment absorbs light at different wavelengths, contributing to the plant's ability to harness light energy efficiently. In chromatography, pigments are separated based on their polarity and solubility in the solvent. As the solvent moves up the chromatography paper, the pigments move at different rates, allowing us to isolate each pigment. Let's review our methodology. This table has the materials used and the key steps performed in the two experiments we conducted. These experiments consisted of the spinach floating method and the separation of leaf pigments. In our first experiment, the spinach floating method, the goal was to measure the number of spinach discs that would float in water in different conditions. Here, we prepared the leaf discs. We then set up the 4 environments for our leaf discs. 4 cups, 2 of which would have a water, bicarbonate, and soap solution, and 2 which would contain only water and soap. We separated 40 leaf discs equally into each cup and then separated the bicarbonate solution cups and a water and soap solution cups individually into dark and bright environments. We observed the number of buoyant discs and recorded the data. Our next experiment was the separation of leaf pigments. In this experiment, our goal was to separate the different pigments in a spinach plant through chromatography paper. First, we prepared our chromatography paper to match our desired length. We then applied the pigment horizontally across several papers. The steps did not require several trials, but we did in order to analyze the best possible separation of pigments. We then prepared the solution which would separate the pigments, put our papers in, and waited. Here's a little time lapse. After 30 minutes, we took the papers out, identified the pigments, and calculated the RF values. These tables show the results of both experiments, including the RF values for our pigmentation experiment. If you'd like, pause now to analyze these results. Now, let's move on to our conclusion. The experiment demonstrated that spinach leaf discs floated as a result of oxygen production during photosynthesis, which caused them to become less dense. Discs exposed to both light and carbon dioxide, from baking soda, began to float after about 4 minutes, with more discs rising progressively between 6 and 22 minutes. This suggests that both light and carbon dioxide are necessary for photosynthesis to occur, as no discs floated in the dark conditions. The delay in floating indicates that photosynthesis takes time to become fully active. Pause now to take a look at our references. Now, we would like to express our sincere gratitude to Professor Murata for his expert guidance and unwavering support throughout the course of this experiment. His knowledge and insightful feedback were instrumental in shaping this project and enhancing its quality. A special thank you to our lab group members, Oscarling Melendez, Mario Alcantara, and Mariana Trier, for their collaboration, dedication, and hard work throughout the experiment. Our collective efforts in data collection, troubleshooting, and setting up the experiment were essential to its success. Working together as a team made this research experience both productive and enjoyable. Lastly, we would also like to thank Miami-Dade College for providing the necessary resources, equipment, and support to conduct this experiment. Thank you for watching our PBL project presentation. Take care and see you soon. Goodbye.